Our first speaker for the end session is Dr. Wisztorski, who's going to talk about mass spectrometry imaging coupled to microproteomics.
Thank you very much. As everybody before me, I will thank the Committee of Organization for this invitation, and I will expose the development we realize inside our labs, what is mass spectrometry imaging, and also all the development concerning identification of proteins from a small amount of tissue. And just to be sure that everyone know what is MALDI. MALDI is for matrix-assisted laser desorption ionization, and on this technique you use a small chemical organic compounds called a matrix, and you mix it with your analytes. And this solution is deposited on a sample plate, and you obtain some crystals inside which you have a mix of matrix and analytes. And after UV irradiation by a laser, you obtain a molecular charge in gas phase, and this molecular will be analyzed by the analyzer part of your mass spectrometer. And this technique is it's a soft ionization techniques. That means that you can detect intact molecules from small molecules, drugs or lipids, to really large macromolecules like proteins. And you can also use it for analysis directly tissue section. And in this case, you apply directly your matrix on the top of your tissue, and the solution will extract your analytes, and you will obtain the molecules in a different part of your tissue. And this is the basis of the techniques called mass spectrometry imaging. It's a technique developed by the team of Richard Caprioli, and you can obtain by these techniques the spatial distribution of a really large panel of compounds, exogenous or endogenous compounds, and you can also use it on for doing a lot of things. Like, for example, biomarker research or diagnosis, and also to understand the molecular mechanism involved in some disease. These techniques, you will first cover your tissue with the matrix by different way. You can spray it, you can spot it, and after you will define an array of points and analyze each point to cover all the surface of your tissue. And on each point, you will obtain a mass spectrum with different signals depending to the position on the tissue. And at the end, you can create a map, reconstruct by the intensity of each peaks on each spectrum you record before. And you can superpose all this information to obtain a correlation map. There is two purposes for which you can use MALDI mass spectrometry imaging. The first one is a targeted approach, and on this approach you know the molecules you want to analyze. It's the case for pharmacological studies. For example, you want to study one drug, the localization of the drugs, and also if there is some metabolites created. And you can use MALDI mass spectrometry for this on organs and also on whole body section of small animals. And on the other end, you have the untargeted approach. It's a more global approach, and on this approach, you don't know what you want to find, and you compare, for example, tumor region to benign region, or control versus treated samples, and by this you obtain a signature of molecules. And in this case, the really important thing is the identification of the molecules, and we will see after how you can realize this. what you also have in mind is that multi-mass spectrometry analysis will generate a great quantity of, a large quantity of data. For example, in this small part of the tissue brain, we realize an image with a spatial resolution at 30 micrometers, and it gives you more than 8,000 spectra. And on all these spectra, you can find more than 500 peaks corresponding to different type of molecules. And it gives a really abundant information, and only on one analysis. And after, you have a lot of different software you can use to better understand this data. And there were a lot of development done on these techniques, and we can now achieve image on spatial resolution near to the size of a cell. Actual instrument can realize some image at 1 micrometer for small molecules. You can also use instrument with a really high spectral resolution. You don't see here, but all the 3 peaks localized here have the same nominal mass, but completely different localization. And by using this type of instrument, you can localize more molecules with really near masses. And you can also have a lot of software you can use to realize, to analyze your molecules. And on this picture, you, for example, have a molecular histology image obtained not on the morphology of the cells, of the shape of the cells, but only by the molecular content of the cells. Okay, great. Now we can realize a really beautiful image, but how to identify the compounds? And there is 2 possibilities. If you work on small molecules, you can realize direct identification on tissue during your analysis by realizing fragmentation on your tissue. But this is not possible for large proteins, and the most common way is to realize an indirect identification. That means an extraction of a whole organ or a whole tissue section. But in this case, there is two difficulties. The first one, you dilute your signals due to the most abundant proteins. If your really interested proteins is expressed on a few quantity of cells, and you also lose your information of localization. That's why we try in our labs to develop some strategy to realize directly on tissue the identification. And the first one was to adapt the bottom-up approach, but by staying on the tissue. We first perform a multi-mass spectrometry image to obtain the localization of intact proteins, and after we deposit on a consecutive or on the same section after removing the matrix. We deposit an enzyme, typically the trypsines, and the trypsin will digest locally the protein, and we can after cover again by the matrix, again realize some image, but this time of digested peptides, and on each point you will obtain a peptide mass fingerprint of local proteins. It's really complex because you have all the protein digest at the same time, and you can, for example, you can select one peak of interest and realize MSMS fragmentation, and at the end obtain the identification of proteins by data bank interrogation. It's working very well, but it's really limited on the number of proteins you can identify due to the complexity of your samples, and also what you identify the most is that it's the most abundant proteins. For example, I extract here a spectrum corresponding to this point after a digestion of the tissue, and what you can observe is that more than 90% of your of the most abundant peaks are coming from only one protein called the myelin, a really high abundant proteins in the brain. And if you want to really understand what mechanism, one biological mechanism is involved in your disease, you really be interested by the identification of all of these small peaks. And to realize this, it's not possible to stay on the tissue, but we develop a technique where we go off the tissue, but still still keeping the information of localization. And we, like before, we first realize mass spectrometry imaging, but this time to perform a molecular histology analysis of our tissue and to obtain some region of interest, and this region of interest is only chosen by the molecular content. And on this particular region, we perform in situ microdigestion of small region. And we, after, extract the peptide obtained by a liquid microjunction, by a liquid microextraction. And this solution is analyzed by nano-LC. It could be analyzed, for example, by nano-LC COP2 high-resolution mass spectrometer. And this time we obtain the identification of thousands of proteins, like you can see here. You have on the left part an example of two discrete regions digest, and we achieve the digestion of a region of 700 micrometers in diameter. It corresponding is not 1,000, but it's more 2,000 cells in this part. And we can achieve the identification of more than 1,600 proteins in these two regions only by extracting the peptide obtained on a tissue section of 10 micrometer thickness. And we also keep the information of localization by realizing this type of experiment. You have the comparison between the two positions, and we only by realizing an analysis, we obtain 200 proteins specific to this position, so position 1, and less than 300 proteins specific to the position 2. Okay, we apply these techniques because here it was on the rat brain, it's not really interesting, and we apply it on the clinical application, and the application we chose was the ovarian cancer, and more especially the origin of the ovarian cancer. It's one of the most lethal gynecological cancer, and and normally we say that the tumor starts on the ovary part, and after some cells go inside the fallopian tubes. But some new hypothesis shows that there is maybe an origin on the fimbria end part of the fallopian tubes. In fact, in this part, in some patients with mutation of BRCA1 or 2, the clinical there is a clinical protocol where for this patient we remove all the annex part to prevent the development of a tumor inside the ovary, and after analysis all this annex, some groups show that some groups show that there is small cellular anomalies appear in the epithelial part of the fimbria end, called the steels and sticks, and that maybe it could be obtain a tumor from these cells, and sorry, from these cells, and after going through the ovarian surface and develop a tumor in the ovary part. We perform a previous first analysis on 4 patients, and the particularity of this patient is that they developed a small tumor in the fimbria part. There are no tumor inside the ovary part, only on the fimbria part, and we perform our strategies first by realizing multi-mass spectrometry imaging to obtain the region of the tumor, and after by making the microproteomics analysis on the tumor part and on the benign part on 4 patients. And we obtain a list of 123 proteins specific to the tumor regions, and by realizing some analysis, data mining using gene ontology, we show that a lot of them are involved in a neoplasia process, and there is also a large part already known to be involved in urogynecological disease and also on ovary cancer. We also propose an hypothetical signalization pathway explaining why on this type of tumor there is an accumulation of the protein p53. And you see here you can reconstruct using the list of protein we identify, we can reconstruct all the proteins in red here is the protein we found by our techniques, and all of this can can interact to inhibit the degradation of p53, and also to activate the expression of the gene TP53. We also realized some analysis, some case study on patients presenting at the same time a tumor in the ovary part and a tumor on the fimbria part. And what we obtain is a really large number of common proteins, and also for the specific proteins on the fimbria part, the profile of these proteins are more invasive and metastasis. And for the ovarian cancer, there is more proliferative profile. Okay, all this clue go to the right hypothesis, and the ovarian cancer maybe start on the fimbria part, and that could explain also why for this cancer there is a really large number of patients that only be diagnosis on a really late state, and really poor on the really early states. Okay, just to conclude for these techniques, you can extract molecules from a restricted region, and it minimizes the dilution that you can obtain from the presence of high abundant proteins. It's also, yes, I have not mentioned, but the first part with the rat brain was done on fresh frozen samples, and the clinical application was done on formalin fixed and paraffin embedded tissue. And it's compatible with both. It's automatic techniques, and you can extract and you can realize what you want with your extract. You can also realize quantification by label-free or label mass spectrometry, and it's also not limited to digestive proteins. But we know that all labs are not equipped with instruments that could realize this type of experiments. That's why we develop another approach where we use something everybody have inside the labs. It's the parafilm, and it's really cheap. You can find it everywhere, and we use the parafilm as a support of the tissue section to realize a manual microdissection. And you can see here, it's really first experiment we realize is not really precise on the for the section, but you can obtain the identification of a lot of proteins directly by extracting proteins of these regions. And what you can also achieve by these techniques, if you cut your section in small square, you need to have a lot of students to realize this, but if you cut it in a lot of square, you can obtain the localization, this time of thousands of proteins identified, and you can realize imaging based on the quantification value of the identification proteins. Okay, I will just conclude by showing also you that you can also realize transcriptome analysis by using multi-mass spectrometry imaging. And to realize this, we developed a modified probe with a tag reporter link with a photo activable linker here. And after UV irradiation, the tag is released, and you can observe it on mass spectrometry. And as you know the mass of your tag, you can really easily detect this on the mass spectrometry. And you can also design different type of probes. And you can obtain multiplex analysis by working with different probes and different tag reporter, and realize, for example, an in situ hybridization on your tissue, and perform a mass spectrometry imaging experiment. And you will obtain the localization of all your reporter. You have an example here of the in situ hybridization of the proencephaline mRNA here on the classical way, and here by the tag mass, by using the tag mass techniques. And it's not restricted to the mRNA or DNA analysis. You can also use antibodies, modify antibodies or modify lectins for analysis all the molecules you want. Okay, thank you very much. I want to thank all my team, the director of the labs and director of the groups, and our financial support and collaborators. Thank you.
How deep are you penetrating in the surface of the tissue?
After applying,
Molecules right on the cut surface?
for which techniques? For mass spectrometry imaging, after applying the matrix?
Digest the matrix, you're getting down into it a little bit, but you're scanning the surface. You're putting the matrix on top of the cut surface.
Yeah, we extract through 12 micrometers around, and what is great is that you can remove the matrix and after perform another analysis by putting, because I don't say, but you can use different type of matrix depending of the molecules you want to analyze, and you can obtain different type of information with the same tissue. It's a non-destructive analysis, because you can perform also also classical histology staining after mass spectrometry imaging, and you extract only a few % of your molecules. And,
Have you ever tried combining LCM with scanning MALDI on the same area?
Not yet, but we have we think about this, and we wanted it to compare with our microproteomics study. We don't want to make a new techniques. It's not. I think it's not at the same level between LCM, where you really study one type of cells, and our techniques is more, you study more the microenvironment of the cells and all the communication of the cells between each, because you have a mix of tumor cells, of stromal cells, and also immune cells you can find.
Thank you.